Hello and welcome to the next instalment of the Oxford Centre for Personalised Medicine Flash interview series, where we talk to individuals playing important roles in personalised medicine in one way or another. Today's guest is Dr. Jamie Ellingford from the University of Manchester. Jamie, welcome. Could you tell us a little bit more about yourself and your background?
Sure. So I'm a research fellow based at the Manchester Centre for Genomic Medicine. And I did a PhD in the same place, which was primarily about how we can use informatic approaches or bioinformatics to high throughput sequencing data sets to learn more about how genomic variation can lead to disease. The skills that I've acquired along the way include programming, bioinformatics, as well as wet laboratory skills in order to investigate the genomic variants that we can prioritize from those bioinformatic analyses.
So what does the term personalised medicine mean to you? And how does your research tie in with the aims and the objectives of personalised medicine?
So personalised medicine to me is about using information about an individual in order to target the care that they receive. And so that could be by defining the most appropriate treatment or intervention strategies. And my work largely is focused around understanding how an individual's genome or particular variants in their genome can lead to specific disease onsets. And one extension of that is being able to use that information to best design how those individuals can be cared for.
Would you like to give us some examples of where your research and the use of genomic data has directly impacted the lives of patients?
So a lot of what I do is directly translational into clinical care. The findings that we have from research studies, once they get to a certain level of evidence, can be directly implemented into a patient's care pathway, whether it's giving them a more informed or more accurate genetic diagnosis, or tailoring their treatment and clinical care based on the genomic findings that we find in our research labs. One really fantastic example of how this can be advantageous for patients was from a case study that we published in 2015. And so this was a family where there was two young children, both of whom had presented to an ophthalmologist with blindness from birth. A whole series of different genes can have genomic variants in them, pathogenic genomic variants in them that lead to these conditions, but a certain subset can also be involved in more syndromic conditions. And so in the young boy from this family, we performed whole exome sequencing. And from that whole exome sequencing data, we identified specific changes in a gene which can lead to more syndromic disease. The gene was a gene called IQCB1. It's expressed in cilia and it's vital for the function of primary cilia. And one of the manifestations of individuals with these types of genomic variants is that late into their teenage years, they also start to develop problems with their kidneys. And usually before the age of 20, they develop end-stage renal failure, which can be a really severe consequence for those individuals. We'd performed whole exome sequencing in this boy when he was age 13. And by implementing these findings into his care pathway, we could get him to a pediatric nephrologist very quickly. He was able to start hemodialysis before end-stage renal failure and ultimately able to find a relevant donor for a kidney transplant, again, before end-stage renal failure. And so all of these are good predictors that he will respond well to that, and really happy to report that at the moment he's doing fantastically well as a result of that.
That's brilliant, thank you. And that's a really good example of how researchers and the clinic can be integrated together to implement personalised medicine. So looking forward to the future then, as a researcher in genomic medicine, where do you see the field of personalised medicine heading in the next 5 to 10 years? And what do you see as the potential challenges of integrating personalised medicine into the clinic?
So when we use genomics in personalised medicine, one of the real challenges is being able to accurately interpret the genomic variants that we're looking at. And in order to increase our accuracy and increase our clarity of reporting back of these types of variants in the clinic, we do need extra information. And so I think it's that post-genome data science, which is really going to explode over the next few years. And that can be additional omic techniques, for example, RNA sequencing, being able to look at specific markers of regulatory elements within the genome and understand how specific signatures are associated with pathogenic variants in certain types of genes. I think that will take our potential to interpret these variants of uncertain significance that we see all the time to a new level, and actually will, for the next 5 or 10 years, be a really exciting place to work in.
That's great. Thank you so much for your time, Jamie.